Hey everybody! So welcome to today's webinar, How to Design and Conduct Challenge Studies for Safer Products and Longer Shelf Life. I'm Adrienne Bloom, Editorial Director of Food Safety Magazine, and I'll be your moderator for today's webinar. So today's presenters joining us from the University of Wisconsin-Madison are Dr. Kathleen Glass, Distinguished Scientist Emeritus, Dr. Kristin Schill, Assistant Research Professor at the Food Research Institute, and Dr. Cynthia Austin, BSL-2 Lab Manager for the Meat Science and Animal Biologics Discovery Program at the Food Research Institute. So I'll rejoin the presenters at the end to help answer your questions that come in throughout the webinar. So don't forget to submit those questions in the Q&A and chat section of the webinar console. And today's event is also being recorded and it'll be archived on food-safety.com.
All right, thank you very much. Welcome to today's webinar, and I am Kathleen Glass from the University of Wisconsin-Madison. I'm an emeritus, but Kristin, Cindy, and I work together at the University of Wisconsin-Madison and collectively have over 70 years of experience with microbial food challenge studies that we want to share with you today. FRI, or the Food Research Institute, was founded in 1946 at the University of Chicago and moved to Wisconsin in 1966. It is the oldest academic food safety group in the United States. We have joined forces with the Food Safety Program at the new Meat Science and Animal Biologics Discovery Building that opened in 2020. FRI is an interdepartmental entity that includes faculty and staff from the departments of bacteriology, food science, animal sciences, medical microbiology, plant pathology, and the vet school to conduct a mixture of basic and applied research. In addition, we have a strong outreach program to assist the food industry and liaison with state and federal regulatory agencies to provide science-based documentation to help answer food safety questions. Cindy manages the Biosafety Level 2 lab at MSABD, which contains meat processing equipment dedicated for working with foodborne pathogens in meat and protein products. That includes smokehouses for fermentations, drying, equipment for thermal inactivation, and incubators for growth inhibition studies. The Applied Food Safety Laboratory, supervised by Kristin, is in the Microbial Sciences building, and that covers pathogen growth inhibition studies for most other chilled foods, shelf-stable processed cheese, coffee, and develops thermal inactivation D and Z values. Her laboratory is one of the few in the United States with authorization and expertise to work with Clostridium botulinum. Before we get started, we'd like to get a little bit better idea of what is the type of needs that you have. So in first question, we have a poll, and we'd like you to answer, what type of study do you need information on designing? Please choose the best answer. A, being growth inhibition studies for refrigerated foods. B, validation on the effect of short-term auto refrigeration or temperature abuse. C, developing thermal processing parameters for cooking or hot fill. D, developing alternate lethality such as high pressure pasteurization or an acid hold or formulation hold inactivation, or other topic, or maybe more than one of these topics. And it looks like we have quite a few people that are looking at more than one type of topic, and keep those answers coming in. We'll be glad to keep this in consideration. Thank you. Now we would like to take a look at the next set of questions as far as what kind of background you have. Sorry about that. There must be a little bit of a delay in this, so I don't know if Vanya can help out with that. Okay. All right. Okay, on my screen, it's going a little bit further. So how would you classify what your degree of expertise is? Do you consider yourself an expert food microbiologist, but just want to get an update? B, do you have a working knowledge of pathogens and the risks? Or C, you are more in the food production and food product design, but have little knowledge in food microbiology? All right. It looks like most people are right about in the middle. And hopefully, what we're going to be able to provide for you today is really going to be helpful for you to go forward. So, the objective of this webinar is to get a broad overview of challenge studies that may be needed for documentation that shows your preventive controls will work as intended in your food safety plan, or perhaps you're reformulating a ready to eat meat product to show inhibition of Listeria monocytogenes. We will only cover the highlights of designing and conducting microbial food challenge studies. We don't have time to go into great depth and do not expect you to be an expert in an hour. For greater details, we recommend a 2-day workshop that is offered each spring by the International Association for Food Protection that will give you more details and experiences. But we will leave you with a list of resources for you to consult and get started. And if you do go into the handout tabs, you will find some of those resources. For this webinar, we are assuming that these attendees have a basic understanding of foodborne bacterial pathogens and factors that affect microbial growth and lethality. Cindy, Kristin, and I will tag team for this presentation. What we want to do is outline the basic information you need to collect before designing a study, how to choose the right microbes, what are some predictive models that can help streamline this process, and considerations for inoculation, processing, storage, and sampling that apply to different types of studies. One of the aspects after collecting the experimental data that is not always straightforward is how to interpret whether the system has passed or failed. There are confounding results, such as when a formulation system is at the border of growth and no growth, or you have erratic growth of spoilage microbes that make interpretation very difficult, and perhaps you may need to invest in additional testing. Now, when people want to come forward for a challenge study, they think it is going to be quite straightforward. Challenge studies do require a large investment of resources, both in terms of money and time, so you want to get it right. You think that all you need to do, though, is contact the lab, get information on the product, inoculate, test, get the report, set the production limits for production, and you're good to go. However, the reality is much more convoluted than that. Product developers should connect with the food safety experts early in the process to get guidance on what the risk might be, if there's going to be any kind of safe harbors, how to control the risk under reasonably foreseeable conditions of productions and storage. But as we will point out many times during this time, you have many factors that you have to consider simultaneously. Food products are seldom homogeneously and they are not sterile. And once again, the indigenous spoilage microbes may interfere with the study, and pathogens can find growth niches within a multi-component food. This can be frustrating to both the industry and the lab running the study. So for those of you who have gone through preventive controls for human foods curriculum, you are aware of the many factors that go into assessing the hazards with foods. These factors include: what are the likely pathogens in the raw ingredients? Do they have a low infectious dose that require lethality step? Do they require growth to cause illness? We need to understand how resistant these microbes are to processing, such as cooking or high pressure pasteurization. Will it kill just the vegetative cells but still allow survival of spores? Speaking of lethality, what process are you using for lethality? Are there factors that are present in the food that can actually accelerate lethality, such as low pH? Or can delay lethality, such as a dry oven chamber that allows greater stability of pathogens during heating? What is the potential for post-process contamination? How thorough and frequent is your sanitation and environmental controls? Could listeria be an issue even though it should be killed during cooking? Are there concerns with extended runs where growth can occur in food residues within the equipment? Perhaps you have a pancake batter that is prepared hourly, but the equipment is only cleaned every 24 hours. Is there any product that holds up in the equipment that could allow growth of Staph aureus or Bacillus cereus that produce a heat-stable enterotoxin that can survive through the cooking processes and therefore cause illness? For the finished product, what is the product formulation? Can it support growth even if the product is properly refrigerated? Not only should we consider the manufacturer's intended storage conditions, but what might also be reasonably foreseeable abuse at retail and consumer levels. Know your customer. What is the potential that the consumer's refrigerator is actually 50 degrees Fahrenheit or 10 degrees Celsius? Or the food may be convenience food that is put into kids' lunch packs that might sit in a locker for a day or two. All of these factors should be considered in the challenge study design. There are two main types of challenge studies: growth inhibition and lethality studies. Depending on the product, you may need to do both. Within each category, the objective may be more specific depending on the product formulation, processing, and storage conditions, as well as compliance with specific regulations. Growth inhibition studies very frequently can be done in a BSL-2 laboratory in a commercial facility, a commercial lab, or an academic lab with relatively minimal food processing equipment, and can be done with the actual pathogens. These types of products might include determining how long a food might inhibit growth of listeria to establish date marking, or whether cultured sugar vinegar might inhibit growth of Clostridium perfringens during extended warm holding of a bean sauce in a food service, or whether a refrigerated meal will develop gross spoilage before toxin production by Clostridium botulinum. For lethality studies, academic and commercial labs can conduct studies to determine D and Z values of a target pathogen for a given food matrix in order to establish a thermal process. Or they may need to validate the efficacy of a hot fill process, or determine the rate of lethality of acid-tolerant pathogens or other formulation components within a food. However, some lethalities may need equipment that's very specific, such as smokehouses, impingement ovens, high pressure pasteurizations that can't be readily replicated in an academic lab. Or they may have equipment that has variable conditions within the unit, or once again, conditions that might be drying or need specific relative humidity. These studies may either need to be using dedicated equipment in a BSL-2 lab, or sometimes use validated surrogates in a manufacturing facility or pilot plant where pathogens are prohibited. Now, make sure that you don't confuse shelf life studies with inoculated pack studies. Shelf life studies frequently only test naturally occurring microbial spoilage on their effect of quality attributes. While knowing the shelf life is extremely important in setting up a pathogen challenge study, you can't use one to substitute for another. About 15 years ago, the U.S. National Advisory Committee for the Microbiological Criteria of Foods, known as NACMF, developed guidelines on conducting inoculated pack studies. In this report, the committee developed a worksheet to collect necessary information needed before designing a study. We provided a blank copy of that worksheet as a handout for your use. As you fill out the form, you will find out that some of the information is known from maybe previous production or prototypes, whereas other cases you may need to do some additional analysis before you can finish it up. Based on this information, we can determine what pathogens will be of concern, if validation of lethality is needed, or growth inhibition study is needed. So information that is needed includes what are the ingredients? Is the product homogeneous, or does it have individual components that may affect equilibration of moisture and pH? If the food includes something like root vegetables that may have residual soil contamination, that can affect what type of microbes are present, such as spore formers. What are the analytical values for individual components and the composite of the food? Specifically, what is the pH, water activity, moisture, and salt levels? Are there clean label or synthetic growth inhibitors added? How consistent do you anticipate the production be? What are going to be the ranges for any of these analytical values? What are going to be your anticipated target or critical limits? Typically, a challenge study wants to test the worst case scenario so that if you do have any process deviations, you are going to have some coverage. What are the preparation steps? That includes time and temperature that the food may be held before a lethality step is applied. What type of lethality is used? Is the product cooked in the bag? Is it exposed to the environment before packaging? Is the packaging reduced oxygen packaging, or is it gas permeable? Even though product developers may be working on a food for many months, many companies come to us without knowing the shelf life of the product under normal and abuse conditions and what kind of changes there are in microbial populations and analytical values. Does the pH naturally drop? These will affect what we might expect to grow and if there are additional control treatments that need to be prepared in order to make a fair comparison. Please consider not only the intended storage conditions, but what is also realistic conditions at retail and consumer handling. As I mentioned before, think about what is going to happen with kids and the consumer's refrigeration. And don't forget to consult any regulatory requirements, such as required validation for foods packaged under reduced oxygen conditions, or if there's other consumer requirements that might be more stringent. Now, once you gather the detailed information, it is time to start choosing the right type of microbe. In the risk analysis, we're going to look for pathogens that are ecologically and epidemiologically relevant and most likely to survive or grow. Now, while the FDA has a hazard guide, we recommend looking at additional resources, including conducting literature searches, looking at CDC data about recent outbreaks, and consulting experts. It is possible that more than one type of microbe might be needed to cover the product and conditions. For most challenge studies, we use 3 to 5 strains of a given species, but there may be cases where more strains may be appropriate. Our Clostridium botulinum cocktail uses 10 strains that includes different toxin types. For Shiga toxin producing E. coli, we use the big 7 S-tex that have been identified in meats. While access to culture collections such as ATCC is relatively easy, the original source of those isolates, whether it's clinical or food or animal or environmental, is sometimes very difficult to identify, much less know anything about their growth characteristics. So ideally, the lab should include isolates that are from outbreaks, whether they're clinical or food isolates, or food or environmental isolates that are not associated with illness, but associated with that particular food product. As an example, Cornell University houses a Listeria and Salmonella collection. Our lab has secured clinical isolates from outbreaks from our local state hygiene laboratory. Unfortunately, in some cases, further distribution may be restricted, but you can potentially even go to other government labs who may be able to ship to appropriate labs. Regardless, it is important to choose appropriate resistant strains, once again, to be the appropriate worst case of what you might encounter in the real world. Strains of E. coli 0157 isolated from apple cider or fermented sausage outbreaks are known to be more acid resistant. And you can actually increase their acid resistance by how you culturing them. Listeria monocytogenes is normally highly salt resistant, but strains isolated from foods such as sausage or cheese that are high in salt are ideal. In general, gram-positive pathogens are more thermal resistant than gram-negative pathogens at higher water activities, but Salmonella is known for its resistance to heat when desiccated. When you're choosing something for a sous vide or a cooked in bag product similar to that, that will eliminate the vegetative cells, but will target spore formers. Now, it's important to, once again, prepare the cultures for greatest resistance that's appropriate for that technology or for testing the effect of acid or heat or desiccation tolerance. You can find details on developing the resistant strains in the literature. We also frequently get questions about substituting pathogens with surrogates. Depending upon the objective of the study and the need for specialized equipment, non-pathogen surrogates may be necessary for lethality studies, but pathogens are always going to be our top choice. However, if we are going to suggest using a surrogate, please choose it wisely. Not all surrogates have been validated for all foods under all conditions. And so it may be necessary to do some additional comparison testing or consult the literature. Now, when you try to narrow down what are going to be those microbes, the first thing that we're going to be looking at is what is going to be the effect of pH and water activity for growth inhibition. One of the things is to, once again, consult the tables and NACMF document or other published literature. These combinations are not exact, but they will give you an idea of what risks you need to be concerned with. In the lower right-hand corner of the table, it suggests that any food with a pH of greater than a 5.4 and a water activity of greater than a 0.94 could potentially support the growth of all major food pathogens. But that doesn't necessarily mean you have to test all of them. However, as you decrease the pH and the water activity, the number of microbes for consideration will be fewer. Keep in mind that this table is only for growth inhibition studies. Where no pathogen growth is expected, inactivation studies may still be required, and this table may not be appropriate. Now, Kristin will take over from here and discuss a little bit more about the use of predictive models to further narrow your choices.
Hi, thank you, Kathy. So George Box is a brilliant statistician, and he once stated that all models are wrong, but some are useful. Hence the reason why we need to exercise caution when choosing which models to use. Models can be useful, but they do have limitations, and therefore it's imperative to understand the nature of these limitations. Some models were developed using microbiological growth medium rather than a food product and can therefore not accurately represent your particular formulation, especially if it contains preservatives. When used correctly, predictive modeling can help direct reformulation of a product or aid in the selection of the number and timing of sampling intervals and the duration of this testing. Models can also help to reduce the number of treatments to be tested based on the predicted growth of the target pathogen or pathogens. If the model fits your formulation and is sufficiently validated, it can be used as documentation for your preventive controls. Many of these models are freely available online, but often require a username and password. Aside from models that were developed using microbiological medium, there are models developed in specific foods, and it's important to pull up that publication that's in the literature to examine what type of food and the intrinsic properties of that food, such as pH, water activity, salt, and the temperature range of that study to ensure it matches with the food or process you are designing the challenge study for. This is an example of the COM-based model, which is an online free tool. In this example, we are modeling the predicted growth of Listeria monocytogenes, Staphylococcus aureus, and Bacillus cereus. You can choose to set the initial level of cells shown on the left, set to be 0 cells, and then input the desired temperature, pH, water activity, or salt into this model, and it will provide an output which is shown on the right. The physical state can either be left in the default mode or can be moved to 1, which would reduce the lag phase and provide a more worst-case growth model. In this case, the default model for that initial physical state was used. In this COM-based model, it's predicting that Listeria will grow faster than Staph aureus and B. cereus at a pH of 6, a water activity of 0.97, and at a temperature of 12.8 degrees C. The data used to generate this model was completed in a broth medium rather than in a food product and does not take into consideration for any impact that antimicrobials may make on the growth of pathogens if they should be added to your formulation. Additionally, you will see numbers that are in gray on either side of the slide bars for each of the type of components or factors. These indicate the ranges in temperature, pH, and water activity that can be modeled. These aren't necessarily the growth ranges, but are the ranges for which data was collected and input into the model. Another online predictive modeling tool called the Pathogen Modeling Program, supported by ARS, is often used to predict cooling of a variety of pathogens in an actual food model. Very small, and to the left, the far left, is the list of bacterial models to choose from that include Bacillus cereus cooling in cooked rice, pasta, or beans, as well as cooling of Clostridium perfringens and Clostridium botulinum in a variety of meat products. On the right is an example of a Clostridium botulinum cooling in uncured cooked ground chicken. To use this model, you need to input the cooling profile as time and temperature into the white box shown that's surrounded by green. The time and temperature can either be copied and pasted into the box, making sure there is a comma separating the values, or you can choose to upload a .csv file containing the cooling profile. Click on Calculate Growth, and the output prediction is shown in the blue box. In this example, the model predicted the growth of 1.54 log CFU in a total of 48 hours. The USDA limits growth of Clostridium botulinum to less than or equal to 0.3 log increase. So either the manufacturer would need to cool the product faster or potentially add an antimicrobial and conduct a challenge study to validate the efficacy of the antimicrobial at inhibiting Clostridium botulinum during an extended cooling. So moving on to sample preparation, inoculation, and packaging. So it's really important to understand your product and know your product and the production ranges for pH, moisture, water activity, and salt in order to select treatments that may represent the worst case formulation and processing condition. The batches submitted for a challenge study can be prepared in a pilot plant or in a laboratory setting so that you can hit those desired analytical targets. For instance, for a growth inhibition study, it would be important to choose a treatment that is on the upper range for pH, moisture, and water activity, and on the lower end of salt. For a thermal inactivation study, a worst case formulation may include one with a lower water activity and higher pH, since microbes are more resistant to thermal treatments at a lower water activity and at a higher pH, or close to their physiological pH. How to inoculate a product with the target pathogen should reflect how that product would likely be naturally contaminated. In a study involving a multi-component meal, for example, that includes pasta, vegetables, and meat, that is then mixed with an acidic sauce, formulated perhaps with an antimicrobial, it would be appropriate to inoculate the higher pH ingredients, such as the meat, vegetables, and pasta, prior to adding the sauce component. The amount of the inoculum used depends on the type of study. For growth inhibition studies, it makes more sense to inoculate with a low inoculum so that you can detect growth of the population throughout the study. But for inactivation studies, you want to inoculate with a high inoculum in order to track the inactivation of a pathogen over time to achieve a specific log reduction, such as a 5 or 6 log reduction of the target pathogen. The product for testing should be packaged in a way that is intended to be packaged, and that may be under specific gas or modified atmospheric packaging, vacuum packaging, or packaged using a 10K OTR film with a specific oxygen transmission rate to allow for aerobic spoilage to grow and spoil a product prior to pathogens producing a toxin in the food, for example, Clostridium botulinum. The temperature at which a study is conducted should reflect how the product will be stored throughout distribution, at retail, and at the consumer level. Therefore, temperatures that represent normal storage for chilled foods, such as at 4 degrees C, as well as mild and moderate temperature abuse scenarios, may be warranted. On the left is a picture of refrigerators belonging to two different consumer types. The bottom refrigerator, which is packed, likely does not have adequate airflow or temperature regulation throughout the entire unit, and it is likely the temperature is not 4 degrees C, but potentially higher. It's reasonable that for any chilled food product, the study to be conducted should be at 4 degrees and 7 degrees C for 1.25 to 1.5 times the intended shelf life, and under moderate temperature abuse conditions at 10 degrees C or 12.8 degrees C for at least the shelf life or until gross spoilage, and certain chilled foods, such as refrigerated dips, may also want to be challenged at 15 to 25 degrees, as they are more likely to be temperature abused by the consumer, perhaps being left out at a party or overnight or in the car. If it fell out of the bag at your shopping or something. It fell out into your car. Shelf-stable foods should be assayed at 27 to 30 degrees C for at least 1.5 to 2 times the intended shelf life to account for consumers storing shelf-stable products for a long duration, perhaps even several years. When conducting a growth inhibition study, the sampling intervals should always include the time 0 as well as 5 to 7 sampling intervals throughout the study. It is recommended to also conduct 2 to 3 trials of the study to account for variability that may occur when using different lots of ingredients and different sets of cultures for each trial. At each sampling interval, at least duplicate or triplicate samples should be assayed over time. If only 2 samples are assayed at each sampling interval, then 3 trials should be conducted. And if the samples are assayed in triplicate, then perhaps only 2 trials are necessary. Some products that show significant variation may require additional samples being assayed at each interval, such as 5 or perhaps 10. Enumeration of the pathogens can be conducted using appropriate selective augers, but care should be given to cells that are injured either by heat or in an acidified product so that an overlay of a non-selective auger may be warranted. It is important for a well-run study to also examine odor and appearance of the product to notice any signs of spoilage, and pH should be monitored at every sampling interval. Duplicate uninoculated samples should also be assayed to examine the levels and changes in background microbiota and spoilage organisms. Throughout the study, it may also be important to measure the moisture, water activity, residual nitrite, Brix, or any other variables that can affect the outcome of the study or impact the outcome of the study. This flow diagram is a very helpful decision tree to support when and which validation study approach is most applicable. If safe harbors exist for your food product, then you can reference those in your food safety plan. If not, then you can look through the literature to find information that will match your product formulation. If there is an exact match, then you can use that literature source as a reference. If not, then you can decide if it's possible to mimic production in a lab scale or pilot scale setting, or whether an in-plant validation study using surrogates would be most appropriate. We have talked a little bit about growth inhibition studies, so now we're going to shift toward discussing lethality studies, specifically thermal types of processes. It's important to design an inactivation challenge study that provides data to support the setting of critical limits, but will also provide data that may be helpful in the unfortunate event of a process deviation. For thermal inactivation studies, it's important to create what are called D values, which is the time in minutes that it takes to reduce a microbial population by 1 log cycle at a specific temperature. For instance, the product would be inoculated with 8 to 9 log CFU per gram of the target pathogen and heated to a specific temperature, and duplicate or triplicate samples would be pulled at 5 to 7 specific time intervals to enumerate surviving pathogens. The survivors are then plotted over time as shown to the right, and a D value is calculated using the negative reciprocal of the slope. These D values shown in the table above are used to calculate the Z value, which is the degree of temperature that change the D value by a factor of 10. Knowing the D and Z values for the inactivation of a particular pathogen for your product can be instrumental in the event of process deviations. To validate a cook step results in a 5 log reduction of listeria, for example, sampling at time 0 and at the end of the treatment may only be needed, but you will need perhaps more replicates or additional trials to gain significant data. Other inactivation studies that don't involve a thermal inactivation, such as the one shown here for acid sauces or salad dressings or acid hold conditions, have several important aspects to consider. But one primary thing to consider is that microbial inactivation or pathogen inactivation as a result of an acid hold will occur slower at lower temperatures than at higher temperatures, and inactivation of the pathogen will occur faster at higher temperatures. As a result, understanding the coldest temperature of the warehouse where the product is to be stored during that hold will be important information, as that would represent a worst-case temperature for inclusion in the study. Enumeration of injured surviving cells may also need resuscitation by plating onto either non-selective augers or by using a selective agar overlaid with a non-selective agar to help resuscitate those injured but not killed cells. Okay, now I'm going to hand it over to Cindy Austin for key points on data interpretation.
Thanks, Kristin. Okay. All right, I think we're back. Okay, great. Thank you. Sorry, there's a little delay there. So now that you've collected all that data, what are you going to do with it? You need to plot the data. And one of the watchouts you need to be careful of, especially in sort of a growth study, is you're taking, in this example, you've got 4 treatments and you've got 10 samples per interval. And once we plated them out, following all of the rules that Kathy and Kristin gave you, it looks like from this data in this chart that no growth occurred through 6 weeks of the shelf life. However, if you plotted out all of the numbers, you would notice that at weeks 5 or months 5 and 6, there's some outliers up there where some growth did occur. And you would have missed that if you only looked at this data using averages. So looking at averages, you might say, I'm good through 5 months. When you look at the data points as a whole, you might actually come to the understanding that your storage should only be held for 4 months. Because of these outliers, you might be sort of on the edge of your formulation, and the products would not be safe at that 5 and 6 month. But again, you wouldn't notice that unless you had played it out or plotted out all of the data. So interpreting your pass-fail, really, you should consult an expert microbiologist. They understand the product and the organisms. You can also look at regulatory requirements. USDA, FDA does give you limits for lethality and for growth, is, for example, for Listeria. Growth is generally considered to be when you see a 1 log increase above the initial inoculum. Now, there are some instances where that's not true. Some other agencies will say a 0.5 log CFU increase for L. monocytogenes is actually considered to be growth. For Staph aureus and Bacillus cereus, we actually look for a 3 log increase before we say that's growth and that the toxin may have been produced and causing a hazard in that food product. And for Clostridium botulinum, we really don't look at growth of that. What we do is we assay for the toxin instead, because growth is not a good indication as to whether there is toxin in the product or not. Rarely it's a simple comparison between an initial and a final population. So if you can imagine, if you have a 6-month shelf life and you're pulling samples at time 0 and at month 6, and you're not looking to see what happened in between, there's the potential that it could have grown and died off. And so if you just looked at the beginning and the end, it may not actually tell you what happened to the product in the intervening months. It's also important to understand that statistically significant differences, so the numbers, may not actually be biologically relevant. So this kind of comes into play when you're talking about increase for Staph aureus. Really biologically important is a 3 log increase, but there might be statistically the numbers differ in treatments, but it's really not important until you hit that 3 log increase. You need to consider variability in your inoculation. Are you getting it mixed well into the matrices of the food, or is it something you're surface inoculating? How are you sampling? Are you taking a composite method? Are you taking a single sample? So it's really important to understand where the bugs are in your sample and how you're going to get them out so that you can assay for them. For inactivation studies, if there is no performance standard, then you need to take what you believe is necessary and incorporate a 2 log margin of safety to be sure that you are making a safe product. Some watchouts, like Kathy and Kristin said, know your product, know what the intrinsic food safety characteristics are and your extrinsic. So what are your pHs? What are your salt levels? What are your water activities? What antimicrobials do you have in there? What potential production issues are happening? Are you having trouble cooling it quickly? Are you having trouble keeping a consistent temperature for heating? Are you having hang-ups in your equipment where you might have potential for like a staff to grow and maybe increase those numbers beyond what is safe? You also need to know what your consumers are doing with your product. So is it a product that is geared towards, say, a kid's lunch, and they are taking it to school? Things happen. They don't actually eat it at lunchtime. They throw it in their locker. They come back the next day and they're like, oh, I still have food here, and they eat it. So what is the potential for that kind of consumer handling when understanding what you need to do to design your study? I think Kathy and Kristin both mentioned test the worst, but be reasonable. But make sure you're using your pHs, your water activities, and temperatures to test your worst possible scenario so that you can use that information to help you with your HACCP plans. Understand if your challenge study mimics how your antimicrobials are actually going to be used in the food. So if it's an ingredient, use it as an ingredient. If you're going to be applying it to the surface, then you may dip or spray it, but make sure that your delivery is the same as how you're going to do it in the plant itself. Heterogeneous substrate interaction, so the equilibration between components. So if you have a yogurt, say with blueberries in it, or cheese with jalapeno peppers, or a meat with a cheese in it, you have to understand that those food safety considerations of the meat and the cheese, or the yogurt itself and the blueberries, or that they will affect microbial growth, and where those two components meet may actually be a totally different environment, and so may have different growth characteristics for the pathogen you are studying. And understand food is not sterile, so there will be indigenous competitive spoilage bacteria that might be in there. They will compete. We know Listeria is not a great competitor, so if there is something else growing in your product, Listeria may not grow, but that you can't count on that indigenous spoilage bacteria to be there every single time. And they can infect and invalidate your results of your study. So here's a trial with a sliced luncheon meat, 2 trials for Listeria, trial 1 and trial 2. Same formulation for both. Trial 1 passed 16 weeks at 4 degrees C, and the second trial, Listeria, started to grow at about 10 weeks. Why? What was going on? Same formulation. And was mentioned earlier, we do want you to look at spoilage, right? So in the first trial, where Listeria did not grow, the spoilage tests show that it started to grow almost immediately and dropped the pH down to about 6.1. In trial 2, the spoilage organisms did not start growing until probably after week 10, pH did not drop, and the Listeria was able to grow in that formulation. So that spoilage delayed the Listeria growth in test 2, but not in test 1. So it's very important to monitor those background microbes when doing these types of challenge studies, because they can prevent the growth of the pathogen you are studying. So if you're choosing a lab, you want to check their qualifications. You want to make sure that they have an expert food microbiologist so that they can assess and prioritize your risks. They understand the microbial ecology of the foods and the factors that affect their growth. They understand the processing conditions, and they also understand the regulatory expectations of either FDA or USDA. They will provide you with detailed reports on the methods, the chemistry, the microbial results, discussion, and can work through any questions and concerns you have with the study. You want to make sure that they have experience with microbial food challenge studies. You want to make sure they have the necessary equipment, if they need a smokehouse, if they need a pasteurizer, if they need incubators, packaging equipment. any analytical equipment and laboratory containment or biosafety practices. They want to be prompt and regular with their communications and good temperature controls used for their studies. Laboratory challenge studies are not necessarily the same as what is used for routine testing. This is just because a lot of specialized equipment is needed for some types of challenge studies, as Kathy and Kristin pointed out. So a commercial lab may not have that equipment in order to do these types of challenge studies. They tend to be in academic laboratories or a qualified internal laboratory. Okay. I think we're going to bring Kristin and Kathy on. Yes, yep. Okay.
So as we're wrapping up, we just want to bring everything together. And we realized that we spent a lot of information trying to squeeze it into about 40 minutes. But what we're trying to impress on you is not, as I said, to be an expert in running challenge studies, but have an appreciation that it's very, very complex. So we have to have an understanding of food production, the microbial ecology within the food, the epidemiology, what are going to be the pathogens of concern, what are the regulatory requirements, how are the consumers going to be handling it. I can't tell you how many times we've had debates with food producers that said, oh, no, that would never happen. We've got really good temperature control. And then we show them the data on really and truly what consumer refrigerators are, really and truly what temperatures are going to be in the deli. And then they become a believer and realize that their system might need to be a little bit more robust than what they thought they were going to be. Also, we want you to understand, just because you run a challenge study, it might not pass. What is your plan B? And if you listen to the experts and say, well, we really don't know. We really think that maybe you want to try one more treatment. Yes, is it going to cost money? But at least if you do it right up front, you may be still closer to market than you would be if you would have to start all over from scratch. And we do also realize that you may have some failures, but if you're going to have some failures at low probability, such as what the slide that Cindy showed, where they had a couple outliers up on the top and everything else seemed to have passed, even though the average had passed, some of them didn't. What that is showing to you is that we are at that edge of growth, no growth. I realize it makes it very difficult in identifying critical control points, but what that shows to you is you need to back off, and potentially you have to do some additional retesting before you become convinced that this is where your lines are going to be. So the reasons for these failures, though, may not be obvious even to an expert microbiologist. We consult with each other and say, why did this happen? And sometimes it is difficult to see because we can't see at a micro level what is happening. But bottom line is you still may need to do some reformulation and maybe a new challenge testing. Once again, we're going to leave you with a series of resources. These are going to be, the links are going to be live in your PDF. Plus, you're also going to have these available as a handouts. I believe that they put into the chat what are going to be the links for the ARS and COM-based predictive models. Those, once again, are not perfect, but they certainly are going to be very helpful in narrowing down what is going to be most likely to grow. Keep in mind that not all of those conditions that are going to be in your particular food product are going to be there, and so you still may need to do a challenge study, but it will help you with that design. So with that, I think we have time for questions, and how would we like to get started with this?
Yeah, thank you so much, everybody. That's a great presentation. And just as a reminder to attendees, before we have our presenters address your questions, I'd just like to remind you that we do love your feedback. Take a few moments to complete our webinar survey when you get the chance. And we have a lot of great questions coming in here. We're going to probably take a little extra time, actually, to answer some of these. And what I'm going to do is I'm going to throw out a question to the presenter who was speaking when the question came in. But if anybody else wants to chime in, please do. All right, so let's start with the first question. This is a little bit more of a basic question, but I think it's a good one to to address, and I'm going to pose this to Kristin. So do you have any guidance on what are preferable intervals of days that should be used for a proper shelf life study? And maybe that's product dependent as well. Could you speak to that, Kristin?
Yeah, so sometimes you may need to, it is product dependent. So you need to know what the shelf life or the intended shelf life is of the product. You want 5 to 7 sampling intervals, but that's where that com-based predictive modeling can kind of help out. When is the organism expected to grow or predicted to grow under ideal conditions? And then you can kind of adjust when you might be wanting to sample. Sometimes you might want to sample early on and have more frequent sampling early on, and then later on throughout the shelf life, maybe more infrequent sampling. So it really does depend on how well that pathogen is predicted to potentially grow in the product and what the product is, what the water activity is. But yep, so I hope that answers the question. Kathy, do you have anything additional? Cindy, do you have additional?
I was going to say, sometimes it requires some experience to know what is likely to occur. So our labs have done so many Listeria challenge studies, and so we got a feel for what is going to be growth characteristics at 4 degrees versus 7 degrees Celsius. And so we look at the formulation and say, you know what, I'm not sure it's going to grow at all for the first 8 weeks. And part of it might be that I'm going to be looking at predictive models. So we might go week 0, week 4, week 8, but then we don't know what's going to happen. And then we might go every 2 weeks, or we might go every week, depending upon what it is. So if there's not, as Kristin said, there's not a single right answer for that. It's going to be product dependent, it's going to be pathogen dependent, and temperature dependent, and taking a look at the experience of the lab that's going to be running it to find out if they've got some understanding of it too. So sometimes it's going to be in days, sometimes it's going to be weeks. So sometimes it's going to be months.
Okay, great. Thank you, Kristin and Kathy. And then the next question I'm going to throw out to Kathy, because you talked a bit about resistance during your part of the webinar. And this attendee is asking, is there also variability in resistance when you're using non-thermal oxidative treatments, such as cold plasma or UVC?
Short answer, I don't know. But I wouldn't doubt that there's going to be some differences. I think what I would do, because our laboratory doesn't work on that, there are laboratories at the University of Wisconsin at Platteville that works on closed plasma, and Dr. Brennan Namira from the Agriculture Research Service, he gave a seminar on cold plasma September of 2024 on the food safety webinar. I would connect with him too. Listen to that podcast and connect with them and find out if he can give you some information. So the answer is I don't know, but wouldn't doubt it.
Well, thanks for sharing those resources from Food Safety Magazine with Dr. Namira, who certainly is an expert on cold plasma, and he would probably be able to better address those questions. But thanks for the info, Kathy. And then this is an interesting question. I'm going to throw it out to Cindy, but anybody else, please feel free to chime in. So for retail refrigerated products, where does the worst case temperature testing conclude? Is it at the retail display case? Is it during home transport? Or is it in the consumer's refrigerator?
I would say typically once the consumer, they're kind of a wild card. So once the consumer gets a hold of your product, all bets are off as to whether they're having temperature control. So once it leaves the grocery store in their car, like I think Kristin mentioned, it fell out in the back of my trunk and it sat there for 10 hours. Is it still safe? Is it still safe for them to use? Or the refrigerator. Again, yes, your refrigerator's set at 4 degrees, but if you have it cram-packed full of product, then it's probably not actually sitting at 4 degrees. So definitely the end user is your issue. I don't know if Kathy or Kristin.
Yeah, I was going to say, if you do take a look, the data from EcoSure was published in 2007, 2008, somewhere in that time frame, where they took a look at different temperatures at retail as well as consumer refrigerators. And you have to think about your particular product, what is going to be the shelf life, and what is it like if it were temperature abused? So if it were in a consumer's refrigerator, and that 1% of consumer refrigerators that are above 47 degrees Fahrenheit, that should be a target, and you want to be able to test it until spoilage. If you have something that would spoil extremely quickly if it were held above 45 degrees, you're probably less likely to test really long at that point. There are some companies that want to do disaster testing too, and so they may be doing testing at 7 degrees Celsius and at 12 degrees Celsius, which is around 55 degrees Fahrenheit, and maybe a day or two at room temperature. But it really will depend upon that particular food and what the spoilage is going to be like. Because, what we said, there's going to be some consumers that will pick it up and say, you know what, it doesn't look spoiled, I'm going to eat it anyway. And there are cases of botulism that have occurred because of that. So. Bottom line is, I would still take a look at EcoSure data for temperature and look at the consumer refrigerator temperatures.
Okay, great. Thanks, Cindy and Kathy. All right, we're going to take a few extra minutes to answer questions here, so we'll go over the webinar timing just a little bit, so please hang on with us if you would like to. Okay, so here's a question I'm going to throw out to Kristin. How often should air plate, APC, or yeast mold be conducted within the staging areas of shelf life study?
Actually, I don't know, actually. I would maybe punt that to Cindy.
I don't know that I would do that, because I think you're talking about a packaged product. So I wouldn't imagine that air quality where you're storing that product is all that important. That's something that you will monitor in the production room for sure as you're packaging the product, to be sure that you don't have airborne yeast, mold, plate counts that are landing on your product.
Okay, thanks for that answer. All right, so I'm going to throw out a question to the group. So I think this is a interesting one, probably something that a lot of attendees are wondering on the webinar. So what happens after a challenge test is failed or fails? Like what are the next steps you should take?
So I think the first step would be to figure out, why did it fail? How did it fail? How bad did it fail? And then either reformulate or come up with a strategy of how to have it pass. That may be adding an antimicrobial if an antimicrobial wasn't there, or using a different antimicrobial or a combination of preservatives. Sometimes we see that formulations fail when a clean label antimicrobial. Well, was it used at the right usage rate that it should be used? You might have to go a little bit higher in usage rate for that particular treatment or that formulation. Or maybe that is not effective. It's not an effective clean label antimicrobial for that target pathogen. We've seen instances where it has suppressed the spoilage microbes, which allowed for the pathogen then to grow and produce toxin in a product. So what works for clean label antimicrobials might not work for pathogen, or what works for spoilage control may not work for pathogen control, and vice versa. So that would be one instance of a challenge study failing. But there are a lot of different reasons why a challenge study failed. The intrinsic parameters of the food product are too high. The pH is too high. So that would require formulation or formulation change to bring down that pH or bring down the moisture. So there's a lot of different reasons why a challenge study would fail. It would be worked out between the expert microbiologist and then the food safety team and product developers of the company.
I think what's frustrating to a lot of people is that sometimes we just don't know what's going to happen. And we try our best to take a look at our experience and the predictive models. But as we said, we're learning all the time, and learning about whether or not one organic acid is effective against Clostridia versus Listeria. Those are things that we don't necessarily anticipate, because some of that work has not been done. That basic research hasn't been done. So those things happen. Yep. And the bottom line is reformulate, retest.
The other point I would add is that the company does not maybe know much about their product, or know what the production ranges are for their product, or there's something in there that we don't know about. And so it's about collecting all of the data. What are the antimicrobials? Is anything in the ingredient statement that's changed from one trial to the next? Lot to lot variation with some of the ingredients. Ingredients that bring in different sets of spoilage microbes. So like I said, it's really tough. So I think knowing your product, knowing your ingredients, and understanding that is a big help.
Okay, great. Thank you all. I have a few different questions that are coming in on the same topic. So I'm just going to throw this out to the group. So when is it considered necessary to conduct a challenge study for the purposes of shelf life estimation or extension? And then also, do you look at a challenge study and a shelf life study as two different things or components of the same process? Anybody want to grab it?
Yeah, sure. I was just waiting if somebody else was burning to take this, but I would say I would consider them differently. Oftentimes, it's important to understand what the shelf life is of the product before we challenge it with the target pathogen. When does it spoil? What temperature does it spoil? Does it spoil? What does it look like when it spoils? Are there gases produced? Like that would be information that's useful up front. So the company, I think, should do a little bit of homework to figure out if they stored their product on the bench or at mild, moderate, or temperature abuse conditions to kind of get an idea of what their expected shelf life would be before the quality goes bad. And then I'd run it through a challenge study when you're actually adding the pathogen to the product to see if that formulation would support the growth or toxin production of that particular pathogen.
As an example, we had two different soups, one being a green based and the other one being a tomato based. They both had vegetables, but they had different types of vegetables that were in there. Even though the pH was the same and the water activity was the same, the antimicrobials were the same, they had different results. And it boiled down to those particular ingredients. We tried to figure out, is there maybe even regional differences? How were those root vegetables prepared? Did they have skin on them or are they just washed? What kind of washing was there? Those can make a real big difference. So when you are looking at the shelf life studies, you need to be looking at not just one lot, but multiple lots with different types of ingredients. And as I said earlier, shelf life study is indigenous microbes. Challenge studies, you purposely add the microbes to see how your system is going to inhibit it. So those are the differences between the shelf life and the challenge studies. One's purpose, the other one is whatever shows up.
Great, thank you. That's definitely great clarification for the several questions we had on that topic. We're going to take the time for one more question here. All right, so there are many different methods for preparing inoculum in a challenge study. How might variations in inoculum preparation affect the behavior and outgrowth of the pathogen in the product?
There are. I think this is a great question and something that we maybe didn't address. So for acidified foods, it's important to acid adapt the cultures, the pathogenic cultures, the cocktail of strains that you're going to be using to make them a little bit more resistant to the acid environment. There's cold adaptation for low moisture foods. It's important to plate them on a plate and collect the cells off of an agar plate rather than growing them and harvesting them from a broth culture, because it's been shown in multiple publications, especially for salmonella, that growing on an agar plate would create a set of cells that are more resistant to thermal processing and low moisture food environments. So that would be something that would be important to consider. Using stationary phase cultures would make them more resistant also. And then when you turn to the spore formers, that's a whole nother ball game, developing a spore crop of the correct heat resistance. Spore crop generation is more of an art as well as a science. So that is just, that's a whole nother topic for a webinar.
Bottom line, it makes a difference.
Yep, big difference.
All right. Great. Well, thank you. This was a great discussion. Unfortunately, that's all the time that we have for questions today. I hope we helped answer couple extra questions by going over a few minutes. I know we had so many other good questions. But if you have any other additional questions or comments, don't hesitate to click the email us button on the console, and we'll make sure to share them with all of our presenters so they can respond directly to you. And then, just please join me once again thanking Kathy, Kristin, and Cindy for their presentations. Now, if you didn't have a chance to fill it out earlier, you'll be redirected shortly to the post-event survey. We do always look forward to hearing how we can make our programs work better for you. And please make sure to visit food-safety.com/webinars for the archive of this presentation to share with your colleagues, as well as information about our other upcoming events. So we appreciate your time, and we hope you found this webinar to be a valuable experience. Have a great rest of your day, and thanks again.